Talk a little bit about photosynthesis. Since we've talked about photosystem 1 and photosystem 2 and two photosystems and everything, what are the relationships between these different photosynthetic processes? All of these organisms can carry out cyclic photophosphorylation. And so in this case, you're talking just about energy. And so light is collected typically in antenna complexes and chlorosomes or chloroplast thylakoids, etc. And that energy then is funneled down into the reaction center chlorophyll, and the electron is popped off, that is, the chlorophyll is oxidized, and that electron is transferred to something like bacterial phaeophytin. This is how it works in purple bacteria. And then, in turn, that electron goes to CoQ, and then down a standard issue electron transport chain. And it's going down the oxidation potential when it does so. It ends up then at cytochrome C, and then the electron goes from cytochrome C back to the reaction center so that it's recycled. And so light goes in, and electrons move around this circuit. The product of this electron transport, though, is you make a proton gradient. And so that's where the energy of the light goes, is the generation of this proton gradient. And the proton gradient is harvested to make ATP. And so light goes in, electrons cycle, and you make ATP. Well, you don't make ATP, you don't phosphorylate ATP, you don't want it. And so the only thing that's consumed in this is light. The question then becomes, if you've got all the energy you need, what's the next rate limiting to the growth limiting thing? It's carbon. So a lot of these organisms that have used a similar or related mechanism to fix carbon. Carbon in the environment, and when we talk about fixing carbon, you're talking about reducing CO2. Taking CO2 and reducing it down into organic carbon, which is typically much more highly reduced than CO2 is. And so you need a reductant. Where does that reductant come from? For the purple bacteria that we're going to talk about next time, this is the same chain that I just showed you. So they can run this way and do cyclic photophosphorylation. But they can also run it backwards to make a reductant. And so you start with a highly reduced compound like sulfide, oxidize it to sulfur, and those electrons then can go backwards through the electron transport chain. And they move backwards because this is so highly reducing. It costs ATP to do this. Because if you're running an electron transport chain backwards, you're pumping protons back into the cytoplasm, right? And that's ATP that you can't generate because you've used the proton gradient for this instead. Ultimately, then, you get reduced cofactor Q, and that CoQ can be used to reduce NAD to NADH or NADP to NADPH. And this NADH is a reductant that's used in the Calvin cycle or reverse TCA cycle or any of those things to suck in CO2 and reduce it to organic carbon. So this is the reductant, and this is how you make it. So the chlorobate, their photosystem, remember, is homologous to photosystem 1. That is the one in green plants that generates oxygen. It has a very highly reducing reaction sign. And so these organisms, they absorb light. They can do cyclic photophosphorylation just fine. But if they want to siphon off some reducing power, the light oxidizes the reaction center. That electron goes to chlorophyll A. It ends up in an iron sulfur center. and it can be diverted. Instead of just going around the process this way, that reduced iron sulfur center can be used to reduce ferredoxin, and the ferredoxin goes to fixed carbon. That's the reductant. But if you do that, you've got a problem, because your reaction center is oxidized. In order for another light to be photon to be harvested, you've got to reduce that reaction center. You have to recycle it. The way the chlorobi do it is from from sulfide, which goes to cytochrome C, and then that reduced cytochrome C is used to reduce the reaction center. So you're recycling the reaction center from the last part of the electron transport chain. So these organisms are doing both cyclic phosphorylation and siphoning off the reducing power here, and the ratio between those two processes depends on the relative energy versus carbon demands of the cell. Cyanobacteria combine these two processes. And so here's cyclic photophosphorylation. This is a different way of looking at the Z skin. So you have cyclic photophosphorylation that they can do. But you can, again, siphon off reducing power up here at this iron sulfur center. These organisms do that at ferredoxin, and so that the electron here can go to Q, or it can be siphoned off as reduced ferredoxin, and that's used then to run the Calvin cycle. But if you do that, again, you've got this issue of the reaction center. Where are you going to get some electrons at an appropriate reduction potential to reduce that? That comes from cytochrome C, but ultimately it comes from an entire new photosystem. This photosystem absorbs light, it plucks electrons off of water, and it feeds those to the same electron transport chain. And since you're running the electron transport chain in the forward direction, you're getting both reducing power and energy from the same process. If you don't need so much reductant, if you're not fixing that much carbon, then you just run this. And so the modulation of how much light goes through photosystem 1 versus photosystem 2 is what titrates the amount of energy versus reductant generated. Does that make sense? I'm not going to talk about rhodopsin phototrophy. That's a completely different thing. I've also got on here, which I'm not going to cover because it's just biochemistry, is some different ways to fix carbon. You've seen the Calvin cycle, the reverse TCA cycle, hydroxypropionate pathway, and reductive acetyl-CoA pathway. This is all just metabolic pathways. There's no light involved or anything. It's just you start with a reductant and some CO2 and you make organics from it. All right. Next time we talk about the proteobacteria, another giant group of really, really abundant organisms.